So we are coming now to a session which we do every year, and we call it Pushing the Limits in Nanomedicine. And we felt that we are proud that this year we have 3 people who will have interventions, and we think that they are of great importance. Each topic is of great importance. So the first talk will be the translation from finding to product by Anil Patri, who serves as the Director of Nanotechnology Core Facility of the FDA and is an expert in this field. He has before been at the US characterization lab for many years, and we felt that he could maybe give us some insight and some hints out of experience. So may I ask you for your intervention?
Thank you, Beat. Yeah. Thank you, Beat. Can you hear me at the back? Yeah? Okay. Good evening. I hope you got your coffee. I'm sleepy. So I thought Beat asked me to present the regulatory considerations for nanomedicine, the translational side. And this is a very exciting time for nanotechnology, as we know. I am personally very excited about nanomedicine, even though we don't see many of the more promising nanomedicines coming through FDA. We see a trickle of these products coming through. So first of all, let me read the statement. The views expressed here are my own, and they should not be considered as the official policy or position of FDA. This is an overview of what I'm going to present in the next 20 minutes or so. Again, nanotechnology at the Food and Drug Administration, a little bit about the Nanotechnology Task Force, which was formed around 10 years ago, and what all products we have approved so far in the last 40 years or so. And we also produce many guidance documents. I'll go through those, and then some research at FDA, and then standards development, which is critical for any translational work. Challenges we face and challenges that you face as you come through with products to FDA. And one area that we thought we can focus at FDA is standards development. And so I'm going to present a little bit about that in the Global Summit on Regulatory Science we have in September. So Beat started the meeting with saying, damn if you do, damn if you don't. And that really applies to FDA. Contrary to popular belief about FDA, this is not what we do at FDA. So the mission of FDA is to protect the public health by assuring safe and effective medical products and safe food for humans and animals. So we have many centers at FDA depending on the products. I'll briefly go through those that are not familiar with FDA. You all know what FDA is, but then unless you're inside FDA, you don't really know what's happening there. Anyway, there are 6 centers: the Center for Drugs, CDER, that looks at the drug submissions. Most of the nanomedicines come through CDER. Center for Devices and Radiological Health. Again, most of the devices go through that center. CBER, Center for Biologics. If you have an antibody or protein conjugates, they go through CBER. Anything related to food, most of them are not pre-market approval, but some components are. So Center for Foods and Applied Nutrition regulates or reviews those submissions. We have a new center called Center for Tobacco Products. And then Office of Regulatory Affairs, or ORA, these are the labs all over the world, and then they go through any any labs and certify the labs and monitor those labs. And then finally, the National Center for Toxicological Research, where I come from, NCTR, is a research arm of FDA. So most of the research is done there. We have around 600 people, scientists at NCTR, looking at different toxicological effects of all kinds of FDA-regulated products. So these are all regulatory centers with research offices and divisions. And each center, depending on what challenges they face, they conduct research. So there are research divisions. But NCTR is completely a research arm of FDA with very little regulatory function. So coming to nanotechnology, FDA formed the Nanotechnology Task Force in 2006, which is composed of representatives from all these centers, all the regulatory centers as well as ORA and NCTR. So the task force is mainly formed to enable FDA to evaluate products, find any gaps, regulatory gaps or research gaps that may be present on any products containing nanomaterial that are regulated by FDA. So this is within the Office of the Commissioner, Office of Chief Scientist, and as I mentioned, it is composed of all these different centers. So there are different functions that the NTF does, apart from research funding, internal research funding, standards development, coordination within FDA, between different centers, as well as international and interagency collaborations and cooperation. The Nanotechnology Characterization Lab, where I came from before, is actually a collaboration between FDA, NIST, and NCI, National Cancer Institute, National Institute of Standards and Technology, and FDA. And you probably heard, and there are sessions about the EU NCL that is being formed. So these are both NCL and EU NCL are try to help products coming through into clinical trials, or they help in the preclinical translational work. So we also coordinate the work with other agencies through NCET and NEHI. These are the working groups within the National Nanotechnology Initiative. Again, this you may know that the FDA does not define nanotechnology. We don't have a formal definition for nanotechnology for many reasons, and we debated this quite for some time, for a few years, and decided not to have a definition. But we do understand that these products are challenging and they're different, but at the same time, the current regulations that we have at FDA, we feel are sufficient. So there are 2 points we consider. The regular NNI definition of nano, where 1 to 100 nanometers in size, where that size brings unique phenomena that enable novel applications, or if they are beyond 100 nanometers, and we have quite a few that have gone through clinical trials that are beyond 100 nanometers in size, but the uniqueness of that size or those properties that the size brings is still up to 1 micron size, is considered as nanotechnology. So these are the examples of nanomaterial in drug products approved for clinical use. These are not in clinical trials or preclinical. These are all just examples, just to give a flavor of FDA's experience with different kinds of products. That includes liposomes, inorganic nanoparticles, protein complexes, emulsions, lipid complexes, nanotubes, nanocrystals, micelles. All these we call today nanotechnology. Many of them have been approved in the 1990s. You can see the dates that they have been approved and the indications they are approved for. So FDA has quite a bit of experience with nanotechnology and nanomaterial. We just didn't call them as such in the 80s and 90s. So this is additional data set for you, and we are still looking at all the submissions that have gone through FDA and figure out what we have approved in the past. And so this is data from almost 40 years of data, more than 350 products that are in clinical use that you can consider as nano. So you can see the majority of those, or not majority, but maybe one-fourth, now one-third, we're still analyzing the data set, are liposomes. And around 18% of the nanocrystals, these are around 15% iron polymer nanocomplexes and so on. If you look at all these, most of these, the polymers, liposomes, micelles, these are all so-called soft material, not inorganic in nature, metals, metal oxides. So the real metals and metal oxides that are novel, new gold nanoparticles and such are here around less than 0.3% of within those. There are a couple of them that came through FDA that are new nanomaterial. But what we have seen is that there is an evolution of these products. So in the last 40 years, we have seen a lot of these drug delivery for different kinds of application, not just for cancer. But what we have seen is most of them were traditional in the sense that there is a drug and a platform, to more recently we see more multi-component material, meaning multifunctional material or targeted with a drug and so on. So they become more complex, as you know, when you characterize material with different components. But again, we didn't see the immense amount of submissions, but we saw a few trickling submissions coming through FDA. So FDA is evolutionary in the sense if new technologies, new techniques come through, we learn from those new techniques and ask appropriate questions. We see some differences in clinic, we learn from those and then ask new questions. So I'm going to touch upon that about the size measurements when I come to that topic. We also approved many devices that contains nano, and then devices at FDA are classified or regulated according to their degree of risk to public. So these are class 1 devices, class 2 or class 3, which are more in vivo. Let's say this is example of a stent, which has a different risk profile versus something such as a glove, which is a class 1 device. And we have approved many of these devices, and these are examples of devices that are approved by CDRH or FDA. So what you see here is the difference is that most of these are metallic or metal oxides, right? So you see silver nanoparticles, gold nanoparticles, titania, silica, hydroxyapatite. So most of them are metals and metal oxides that are used in devices, so that's the difference. But again, we have a lot of experience within the Center for Devices in reviewing and approving these material. Again, a very brief overview of the submission process. Again, this is a translational work. Many of you are working in the preclinical stages and trying to develop concepts, hopefully eventually getting through the clinic. Depending on what it is determined by FDA when it comes through. We have INDs, which are investigational new drugs. If you have a drug with a nanoparticle going through an IV infusion, most likely it is coming through an IND submission. The IDEs, as I mentioned, these are devices. They go through investigational device exemption. New drug application is beyond the clinical trials, phase 1, phase 2, phase 3 clinical trials. They go through NDA. The BLA or biological license application is similar to IND, just except that it is a biologic. And abbreviated new drug application or ANDA is usually used for generic drugs. So when it comes to nano, the regulatory considerations are no different from any other small molecule drugs. And we went through all this, and the nanoproducts are not treated differently than any submission that comes through FDA. So you have to look at the regulations and guidance documents on that. And we have many guidance documents. I'll show you a few. And so we follow this 21 CFR 314.15D, which requires a full description of physicochemical properties or characteristic stability for drug substance. So for nano, this means the methods you use are slightly different. And we presented this when I was at NCL about the different kinds of characterization methods. So for a small molecule, you would do NMR, maybe mass spectrometry, HPLC, GCMS. But when it comes to nano, you would use maybe dynamic light scattering, static light scattering, electron microscopy. The methods are different, but you're still defining the product. The particle size, the crystal structure, in some cases, the surface area, surface coating, the density of the coating, the quantity of the coating, what is the homogeneity, heterogeneity of the coating, how much of these drugs are free or encapsulated if they are drug-containing nanomaterial. So these are all relevant questions that one need to answer. The identity, the strength, quality, purity, potency, bioavailability are all in this document. And the manufacturing processes and controls and analytical procedures, including alternative procedures. So there was a question about in the previous session as to what method do I use? Dr. Badenholz posed that question. Now, it depends on what you have. And if it is liposomes, and he's one of the leading experts in liposomes, he knows better than any others in terms of how to measure those. The main question is, whatever you're measuring, are you detecting or can you detect any differences if the product changes? In other words, if you have a 100 nanometer size liposome, an average size with dynamic light scattering between 60 to 120 nanometers, if you have more of the 60, 70 nanometer component in the liposome, can you detect using dynamic light scattering? Does it matter? Does it clinically matter? And these are the questions that the sponsor need to ask and answer. So again, there are only known maybe 1 or 2 standards in dynamic light scattering at ASTM or ISO, but those are not sufficient. These are batch mode measurements, and sometimes you need to have flow mode measurements, whether it is field flow fractionation, size exclusion chromatography. But beyond size, the surfaces do matter. I have my former colleague here, Marina Dobrovolskaya, who does a lot of work in immunology, and the surfaces do matter in terms of immune recognition. If you have coating or lack of coating, will completely change the protein binding, opsonization, and biodistribution. So size is only one factor that you need to monitor, but the surfaces are quite critical when it comes to these nanoparticle technologies. About the guidance, so many years ago, 10 years ago, there were no guidance document relevant for specifically nano. For example, there are liposome guidance documents, there are guidance documents in polymers and other things before we call them nano. These are the specific guidance documents that we came up with in the last 10 years. So we issued 4 final guidance documents and 1 draft guidance documents. Again, here I didn't list the liposomes. There are additional guidance documents on liposomes and devices that one can go through, and you can go to this website, and then you can download the guidance documents from FDA. And there is 1 draft guidance that is coming through from Center for Drugs about the nanomaterial in drugs. There is a final guidance as to considering whether an FDA-regulated product involves application of nanotechnology. And then the final guidance for industry on safety of nanomaterial in cosmetics, and so on. So we have quite a few guidance documents that we came up, and it takes a long time to produce these guidance documents and go through reviews, internal reviews. So in terms of research, we do a lot of research at FDA, and even I did not know, and most of us don't know about internal research at FDA, and we don't talk about most of these. Maybe dozens of research projects. These are only 36 research projects funded by the Nanotechnology Task Force in the last few years. But then there are many dozens of research projects in each center, depending on the products they see, and they're not even listed here. So we do quite a bit of research. We have a lot of equipment. We buy new equipment because dynamic light scattering may not be the only method. You have a Q nano. You may be, different vendors may have different kinds of particle size distribution. There are new products that are coming through. Every 6 months, I see a new particle size measurement technique. So if a sponsor comes through with a new instrument, and we cannot deny that you cannot use this instrument. So we have to understand what that instrument does and how it works and how it assures the product safety, product quality. And so we go through the immense amount of new products that are coming through in terms of instrumentation, and then we understand how those will influence our decision-making process. So we train regularly at FDA in nanotechnology. Every 6 months, reviewers go through this training. So some of the projects within FDA include nanomaterial detection. The characterization is very critical, even though the safety and efficacy evaluation is produced by sponsors. One of the main challenges is a characterization. How do you know from a batch-to-batch consistency perspective the quality is maintained? And there are many research projects in that area. Obviously, in vitro effects, immunological effects, cytotoxic effects, as well as in vivo studies on safety. Some of them are GLP studies. We do small animal studies, non-human primate studies. NCTR has a non-human primate colony. As needed, we conduct those studies. Sunscreens, devices, diagnostics, a lot of projects on food contact material. And the instrumentation, as I mentioned, we have quite a bit of instrumentation at FDA, both at the NCTR campus as well as the Whitehall campus. And this is really challenging. Because of lack of standards, there are only few standards that are available that we obviously need more of these. And so I'll briefly talk about those. So the challenges include, we need additional research to bridge a change in material attributes and analytical methods and how those attributes influence the biological disposition, safety and efficacy, and product quality, product performance. So we need to look at those. And then the detection, identification, quantitation of complex matrices, such as blood and tissue samples, especially if they are not labeled. And then, as I mentioned, about the surface coating of the nanomaterial, which is very critical beyond the size measurement and lack of appropriate standards. So FDA recognized, or CDRH recognized only 4 standards, and these are the list of 4 standards recognized by FDA. I know ISO TC229 has close to 50 standards, and ASTM E56 has around 20 standards. So out of those, only 4 were recognized, and we certainly need more. So FDA is heavily involved in standards development. We have around 250 staff participate in different committees and standards committees, and we recognized around 100 standards, but only 4 of them relevant for nano. So we met last year in Parma in Italy. Most of the regulatory agencies were there, the EMA, EFSA, ECHA, the JRC, and all the regulatory agencies, as well as some of the research agencies and standards developing agencies, to discuss about what kind of standards the regulatory agencies need. And came up with a list, and this is a list of reference material that were asked by the regulators who participated in these discussions, as well as the documentary standards that are needed. So these are characterization of material in pristine state, and then these are the list that we came up with the characterization of nanomaterial in complex matrices. So we plan to have another meeting on this. It's called the Global Summit on Regulatory Science, or GSRS, where we are going to expand this discussion and then bring others into the discussion, including academia and industry. And so I highly encourage you to participate in this. This is in September at NIH campus. And so we are going to discuss mainly on the nanotechnology research and standards, applications and standards. So I personally feel that nanotechnology has a lot of promise. We didn't realize the promise as much as we should have. But these standards will enable the commercialization or clinical translation of these products. So in summary, immense potential exists for nanotechnology and nanomaterial, for nanomedicines and diagnosis, detection, and treatment of disease, early detection of disease. And we have seen many of those presentations today. The key is quality and reproducibility for clinical translation. So you have to have reproducible data, which is not easy in nano compared to small molecules. But if there are changes to the product that change the clinical outcome, this is a most challenging component. But I still feel that this nanotechnology has a lot of promise. So again, as I showed you, FDA approved many nanotechnology-based drugs and devices, so we have quite a bit of experience in that area and issued guidance documents, and we're issuing more guidance documents as relevant. challenges do exist. We understand that there are many challenges, and we are not naive in that area, especially when it comes to multifunctional material. If you have antibodies with an imaging agent and a therapeutic, the heterogeneity and homogeneity of those cases is quite challenging to produce reproducibly. And we are engaged in collaborative consensus standards development with other regulators from Europe and Asia and elsewhere. And we talk to other standards agency in Europe and in China, Japan, in Korea. So we are coming together in that area to enable commercialization of any new technologies. So again, the Nanotechnology Task Force is really responsible within FDA to discuss these issues and come up with whatever is needed for clinical translational commercialization or even training and research activities within FDA. I would like to thank their involvement in this. And thank you all for listening to me. Happy to answer any questions.
Thank you, Anil, for this impressive talk. Who would like to ask a question? We have 5 minutes for first questions, and then continue, and in the end have another 20 minutes. Tore. Push the button.
Is this better? Yeah. You mentioned that nano products will be handled the same way as all other drugs. I wonder about the so-called theranostics, products that are expected to be used both for therapy and imaging. Have you had any such cases or discussed how we would handle such a product?
Yeah, so probably Frank Weichel might have discussed this in the previous session on theranostics. So when these submissions come through FDA, we decide on, there is an office of combination products that decide on where these things are going, whether they are drugs related, so they go to CDER, or CDRH if they are device related. In most cases, such complex products are usually you have one center that is leading, and the other centers are involved in the review because you have expertise between the centers. So I don't exactly know if we have seen such products, if that is a question you're asking, if you have seen those products. But we collaborate between the centers to answer any questions the sponsors may have.
The question is really if you need to show that it's better both for therapy and for imaging than products on the market, for example.
Can you please repeat it? Sorry.
If a company is applying for a theranostic, do they need to show that it's better for therapy and better for imaging than products on the market?
Better for therapy, better for imaging. Frank?
If I may, actually, you can get a product approved under non-inferiority, if that helps. So it's a consideration. If you gain anything, certainly it perhaps is a market advantage if you compete against others. But from an FDA point of view, we are not necessarily involved in determining how big or how small the market is. We have to make sure it's a product worth exposing human beings to, that human beings have a benefit from it, and that has a reasonably safety profile.
Alberto.
Anil, let me ask you about, I have actually two questions. The first one is regarding the recent successful outcome of the Celator product of a co-encapsulation of daunorubicin and cytarabine. I was wondering, when the potency is the result of two APIs within the same nanoparticle, how will you really regulate the potency of this product? Is it on what basis? And do we need a new standard unit for that? And my other question, which is related to that, has to do with targeted products. When you have a targeted product, also you have a ligand. And I wonder if you refer to the ligand as also as, not as an ingredient, but as an API, or how do you control the accuracy as to which the ligand in each batch is presented?
So I guess the first question maybe Frank can answer. I can probably come to the second question.
Well, let me
Frank.
start with the second question, but
so I need
well, you have to show
mainly
batch-to-batch consistency, right? You know that. Batch-to-batch consistency has to be demonstrated, and there are certainly parameters that you have to match.
Yes. But when I treat patients, if there are two APIs, how do you really deal with that? Is it, I mean, you really have to give to the potency of this preparation probably a new standard unit that has to be the product of the two APIs. So I think this is relatively something new that I have never yet faced in the clinic when I give a product.
So again, this is my personal opinion, so you cannot hold FDA responsible for any of this. So you do give, and you're a clinician, and you do give combination drugs, right? You don't treat cancer usually with one drug. You do give multiple drug therapies. So when things come through FDA, clinicians look at the data, and then they review those accordingly. So if you're talking about the specific product, for example, if you have co-encapsulation in a nanomaterial, so you have to develop methods to show product consistency and then the potency of that product. And if any change in the combination would change the potency of the product, which may change the safety and efficacy, that is a challenge that you as a sponsor will face. Right, and then coming to the targeting.
But I'm coming back. How do you express the potency? I mean, when you are injecting two separate drugs, you control the dose of each drug. When you have a co-encapsulated material, you need, one drug drags the other one inside the patient. So are you going to create for these nanoparticles? And you said very well that people are going for more complex systems. So you need, I think, a new way to express potency of these products.
So yeah, so what I'm saying is the potency is a combination. That's what you're going after. And it's not a single drug, that you're going a concoction of drugs in a nanoparticle formulation. And then so we review those accordingly as a combination, not as a single drug, as a combination. The main challenge I was talking about is to make sure that the release profiles and having the same amount of these drugs and then characterize those, that is the main challenge that you as a sponsor has to bring.
I guess we shall now go to the discussion, and thank you for the moment.
Yeah.
And then